I don't want to stress my voice. I think I'm getting worse. Alright, everyone ready? So today is chapter 3. Quickly settle yourself. Do you think should I lock the door?
Yes.
Even worse, still knock the door and then ask me to open it. Should better not. Alright, chapter 3. This is the continuous chapter for the last one. The last one we also talk about the bacterial structure and function. So today we continue to talk a little bit more about that. The problem is that I cannot see. Alright, so I have to see it this way. So we continue with the lipopolysaccharide, the LPS. Yeah, we have the very last few slides. In last lecture, we talk about the LPS, how LPS is a virulence factor, and how it binds to the toll-like receptor and cause inflammations in host cells. And today we will also to look at the structure, what is the three components present in the LPS, and then we talk about the extracellular layer, other than the cell wall or cell membrane, what other extracellular layer on the surface of cells, and external structure. External structure include fimbriae, villi, and also flagella. So these are the three external structure. And then we go into the cytoplasm. There's a lot of component in the cytoplasm. And then finally the endospore. Okay, so this is the sequence of the content. For learning outcome, it's very straightforward. First of all, as last time, that you need to be able to describe the structure from the materials, whether it is extracellular or intracellular. Both that you need to know what are the structure and also know their functions. Secondly, you need to differentiate, meaning distinguish the differences of the intracellular, the cytoplasmic component between the prokaryote and eukaryote. You may not need to know what eukaryote have because eukaryote have much more complex intracellular organelle. So you may not know, but you need to know the prokaryote intracellular organelle. What are they? Are they different from the eukaryotic organelle or not? Lastly, then endospore is part of very important, one of the stages for the bacterials, and also is very important for when we study about the bacterial infections. So we study about the structure of endospore as well as how it's been developed. Okay, get yourself ready? So we start. Now, last time we talked about LPS. What do you remember about LPS? LPS only present in gram-negative bacterials. It is anchored at the outer membrane of gram-negative bacterial. So it is very unique to gram-negative bacterial, but not the gram-positive bacterial. So this is the gram-negative bacterials. Always remember, has a very thin peptidoglycan. The cell wall is very thin, and is like sandwiched by the outer membrane as well as the cytoplasmic membrane or inner membrane. Here, pointed out here, this LPS is actually coming out from the outer membrane and then is protruding outward on the surface of the outer membrane. And those bacterials, gram-negative bacteria who has this LPS include the Escherichia coli, Pseudomonas aeruginosa, Haemophilus, influenza, Helicobacter pylori, Klebsiella pneumoniae, and Salmonella enterica. So those are I think it's very, very common gram-negative bacteria, and they all have their LPS. And some of them is also, I think all of them are pathogenic in certain stages. Now, this is something we knew that we have not talked about. What is new is this, the component, the three component of the LPS. First, O antigens. And then in between this and this, the lipid A, I think we briefly mentioned in our last lecture, lipid A and the toxin. And between O antigens and lipid A, we have a core. Here they divide core into outer core, inner core, but we can actually collectively call it as a core. And O antigens and the core, they are polysaccharide, meaning that they are, each of this structure here is a sugar unit. They are the carbohydrate, and they make such a sugar unit, make it into a chain, and it's called polysaccharide. It's a polymer. Here, lipid A, different from the other two, it is a lipid. And because why it has to be a lipid? Because poly A is the one that at the bottom that embedded inside the membrane. So it has to be similar like the phospholipid. Otherwise, it cannot be inserted in the phospholipid, the lipid bilayer. So remember, polysaccharide, hydrophilic, and this is hydrophobic, which is a lipid. There are three components. We start talking about from the O antigens. O antigens is a glycan polymer, meaning that glycan is meaning the sugar. So it has 2 to 6 sugar, making it up. However, so far there is identified more than 20 different sugar units that in the bacteria, in the gram-negative bacteria. So the combination of these 20 different sugar units can make up that different O antigen, more than I think 100 serotype of based on the O antigens, right? And highly antigenic. What is antigenic? Immunological specificity, or immunogenic or antigenic. Immunogenic meaning that it will cause our immune response to be activated. That's immunogenic. Antigenic, coming from like antigen. So O antigen itself is antigen. Why it is antigen? Because once it's under our body, our immune system will produce antibody against it. That's why it is called antigenic. It's allowed the antibody being produced against it. And interesting is that you have the bacteria that, let's say you have a mutant, the one that without the wild type, right? This is the membrane, and you can see this is the LPS. Let's say this is a lipid A, this is a core, this is the O antigens. So there's one wild type, and they have a mutant. The mutant that is without the O antigens. What they surprise them is that once they actually culture on the agar plate, what do they see is that the wild type, they have a very smooth surface. However, the one without the O antigen, which is the mutant, they have a very rough surface. So it's very easy to distinguish between the smooth and rough. And more important, why they know that this O antigen is causing a problem is one of the virulence factor. And the reason is because when they actually removing the O antigen like this one, right, somehow that's tested in E. coli, Salmonella, removing the O antigens, right, you have partial loss or loss of the virulence. That's why they are not like creating very, not as virulent as the wild type. And what they also found that if they are removing the O antigens, they also losing partially or fully losing that the attachment on the whole cells tissues. Okay, some of the other functions of the very long O antigens include it protect from being engulfed by the whole cell phagocyte. What does that mean? If this is the presence of this, right? they actually can have to be not being recognized easily by the phagocyte. Phagocyte here, about in human, the phagocyte is referred to the macrophages, dendritic cells, which they can engulf the bacterials. However, in the presence of O antigen, some of them is very long, it actually protects the cells and somehow not being recognized by the phagocyte, by the macrophage. So it can actually protect the bacterial cells from engulfment, from the phagocytosis by the macrophages or dendritic cells. And also, of course, resistant to the hydrophobic antibiotic. Why? Because some of the antibiotic, such as the kanamycin, they are hydrophobic. And you know that O antigen is made up with O antigens. They are a chain of sugar. So sugar as carbohydrate, they are highly hydrophilic. So because they are hydrophilic, they resist to the hydrophobic antibiotic. And they also become of the antibody. I think the entire, not only O antigen, the entire LPS also limited the access because they are actually very packed, very packed on the surface of the bacterial cells. So they are actually sort of like barrier for anything is coming to passing through the outer membrane. So it is limited the access of molecule, especially the antibody, to the bacterial surface. Okay. Now we come to core. Core, same as the O antigen, is polysaccharide. And here is showing you this is O antigens. The core is around this one. It's connecting the lipid A and the O antigens. They will say this is also sugar. These are the sugar. In addition of the sugar, similar to the O antigens, and they have other things, which is, for example, they have this phosphate group. So the phosphate group also make the core as very negative charge. And in addition of the phosphate group, they have this one. What is this? This is the ethanoamine. Ethanoamine is one of this. And the most important is this. This is called 3-deoxy-D-minooxyurosonic acid, or we call it keto in short. So keto is something that is important because it's linking the core and the O antigens with the lipid A. So this linkage is called ketosidic linkage. And this is quite conserved in a way that it's also very unique to the gram-negative bacteria. So it can be used as a marker to prove the presence of the LPS, or to prove the bacteria that they found is actually gram-negative bacteria, because this is very unique that it's present only in the LPS and in gram-negative bacteria. All right. Lastly, we have lipid A. Lipid A, as you can see from the diagram, the structure is very simple. It has this one. What is this? This is the glucosamine. You remember you have studied the N-acetylglucosamine. This is NAG. It's one of the unit glycan monomer in the cell wall, right? NAG. It's also present here, but it is formed as a dimer. So it's formed as a dimer. In addition of that, you of course has a phosphate group at the linking with the glucosamine dimer. And bottom here, we'll have 6 to 7 fatty acid attached to these two dimer, the dimer here, and here say that out of these three, this is the most conserved component among all three, because it doesn't actually it's because it's attached to the phospholipid, so it has to be hydrophobic and it has to be fit into the membrane. So it's therefore it's constrained. There is a structural constraint for this particular component. You cannot change too much. All right. Here say that it can be recognized, that we in our last lecture we actually already mentioned is very highly virulent, and because this lipid A can be recognized by the toll-like receptor 4 and also the MT2, right, co-receptor, and because of the recognitions that is trigger our immune system, the innate immune system, to actually become inflammatory stage. A lot of cytokines being released because of that. And the cytokine release and even further activate the production of immune cells. So it's actually accelerate the inflammations. All right. So if you're clear about that, let's move on. This one is extracellular layer. You heard about capsules, slime layer, and S layer. They may not be paying much attention unless when you draw a bacterial cells, normally you don't draw these three components because they are actually on the surface. You may or may not notice this unless you stain the cells strongly, then you can see. For example, this is the staining, which you can see clearly. This part, right, is the capsule of the cells. So that, of course, this is a chain of cocci, and you see that is a capsules. Something that is a bit fuzzy, that present on the surface of the cell. It can be it's not very thin, actually it's very huge. And this look like something that carried by the bacteria. So people wondering what are they? So they start to actually investigate what are they, and they found they are extremely also, one of the components can be resistant to the whole cells defense system. So first of all, we look at the capsule and the slime layer. What are they? They are called glycocalyx. Glycocalyx, simply, they are a lot of these polysaccharide or carbohydrate polysaccharide. Sometimes they can be polypeptide, or it can be both, a mixture of the carbohydrate and also the peptide protein. So it can be mixture or either one. So they are glycocalyx that they actually release, secreted by the bacterial cells, and they form a mesh of network on the surface of the cells. So you have a many layer. If you're actually looking at the envelope, you have the inner for gram-negative inner membrane, periplasmic region, outer membrane. It's not even the LPS here. So on top of the LPS, they have secreted by the bacteria to have this capsule. So they have this. And even this electron micrograms clearly show that this is a cells. I think they may be dividing, but outside here, this is a capsule, very clear, right under the electron microgram. Now, the difference between the slime layer, basically they are the same thing, slime layer and capsule. Sometimes people use even very loosely using these two terms. However, if you want to precisely define them and distinguish them, slime layer is loosely associated with the bacterial cells and can be easily washed away. So it's something very loosely organized and a little bit thin. The capsule, on the other hand, is highly organized and it's attached tightly to the bacterial cell and has a very definite boundary. So you have a very sharp boundary and is highly organized. On the other hand, slime layer are not very organized, it's loosely attached, right? It can be gone any times. So these are the difference. But despite the differences in the morphology, both of them are made up with the glycocalyx. Oh, that's better. Okay, now we're looking at what were we looking at? Capsule. So we talk about the capsule first. Capsules, as there are three examples that this bacteria may have the capsules. Bacillus anthracis, Streptococcus mutans, Klebsiella pneumoniae. Now, these three have the capsule, but their capsule for the first two, they are polysaccharide. right? They are carbohydrate, a lot of carbohydrate. They are hetero, meaning different carbohydrate. Homo, they meaning they have the same carbohydrate make up with the capsules. Bacilla anthracis is slightly different because they are not made up with the carbohydrate, but they are made up with the peptide. So this peptide called polygamma glutamate are making the capsule for the Bacilla anthracis. And what is the functions of the capsule? Is actually attach cell, bacterial cells to the solid surface. And secondly, it protects the bacterial cells from external environments, meaning that because the capsules can retain the water, so when in the external environment, when in very, very dry condition, dehydrated, but you still have the water being provided by the capsule. So it's protecting the bacterial cells being dry up. All right, it also can consider is a virulence factor. Such an example like Streptococcus pneumoniae, when it's encapsulated, easily killed by the macrophages. So we know that Streptococcus pneumoniae can cause the pneumonia, right? So when you have the Streptococcus without the capsule, it is it is not virulent, and it can be easily killed by the macrophages. However, if they contain the capsule, and it's very infectious. Therefore, from this kind of experiment, they know that with or without the capsule can determine whether it is virulence or not virulence. Now, this is another example. Streptococcus pneumonia is one. The other example is the Bacillus anthracis. Anthrax, you heard about anthrax? Yes, I think from 911, around that period of time, they used anthrax. This kind of bacteria, anthrax bacteria, has been used as a bioweapon. You can actually send somebody a letter, a document, but this document actually already paint with the, let's say it's the Bacillus, is very commonly used as the Bacillus anthracis, is paint on the letter, even on the not the cover, inside the letter, and then somebody open it, and if they actually touch, they may infect them. But normally Bacillus anthracis will infect the cattle, so like cow or other animals. Not very common for human, but it can be transmit from animal to human or from the environment. So it's possible. Now, what's so special about this? It's so special for one thing, but before we go for that, it said that this is a very bad example. I should make this italic. All the bacterial species name has to be italic. Cannot be like this, all right? Has to be italic. So it does not produce capsule when they are grown outside the cattle. Meaning that when there is no harm, they are not being threatened. When they are living in the environment, they do not produce capsule. When they are inside, right, inside the cattle, then they produce the capsule, meaning that capsules is allowed to protect the bacterial cells from the immune system. So this is a light microscopic picture of the Bacillus anthracis, and you can see they are rock-shaped. Is gram positive or negative? When you see this color? Positive, all right? Bacillus is positive. And you see inside these cells, what is that? It's a hole. What is this hole? They are the spore. So they are bacillus, they can form the endospore. And they form the endospore. Endospore allowed them to be placed in any very constrained, very unfavored environment or conditions, they can survive. So they have a spore that the spore can last for many months, very long time. That's why it's a very good bioweapon, because you can put it in anywhere. the spore can survive for many, even years. So when they actually go into the animal, what happens? They start to grow because inside the animal they have the nutrients, sugar, whatsoever, also water, a lot of water. So they start to actually germinate. So they start to grow. And they notice that when they start to grow, they become the vegetative cells, which is a rock shape, very long. Then you found them, they actually covered by a so-called PGA. What is PGA? It's polygamma deglutamic acid. You know, the polypeptide that they actually produce as a part of the capsule. So this is how they actually make it off. So this is the capsule with or without capsules. With the capsule, they can survive inside the animal. Without the capsule, they can easily be engulfed by the macrophages. For capsule staining, I think you're going to have a chance to stain the capsule. Do you? Yeah, I think so. You will have a chance. Actually, you're going to do this kind of experiment. And when you see this one, there are two types of stain. This is sulfonin, which is supposed to stain pink. What is the halo shape?
Right.
The halo shape, which is the white color here. Same thing, you have the white color here, the halo shape. This is indicating they are the capsule. And why it's a capsule? Because capsule is especially for capsule, they are very organized in a way, very tightly packed. And the stain, sulfonin here you use, and even the Indian ink, I think this is Indian ink that you use, here cannot penetrate into the capsule. So therefore the capsule not stained. What is being stained is the cells, the bacterial cells, and also whatever and the background. Because you actually apply a lot, you didn't wash off the background. So the background will stain very densely and leaving the capsule unstained. The reason not unstained is because the mesh is tightly packed and that ink or dye or stain cannot penetrate in the capsule. So therefore allow us to identify the capsules. Okay, slime layer. As I mentioned, slime layer is exactly the same thing as the capsule, but the way they arrange and organize are different. They are also glycocalyx, but they are loosely attached to the cell surface, and they play the same role as the capsule and prevent loss of nutrient and water because they can actually keep the water in the slime layer, and they can actually attach to the surface, also very important because they can attach to the whole cell's surface. And also this one, biofilm productions. We are not actually go into the biofilm now, but later on in the, I don't know which chapter, we're going to talk about that. This is a very important kind of like structure that created by the bacteria, a population of bacteria called biofilm. And this is very virulent. It's not easily being destroyed if the bacteria inside your body form a biofilm. That not easy. It's like a group, like a population could be 100 to 1,000 of bacteria actually forming a colony. So if I use a colony, something like colony, stick together and they form a biofilm, right? It's not easily being destroyed. All right. Now, this is an example of the slime layer. How you see that is the gliding, we call the bacterial cells here called Stigmatella aurantiaca. So these bacteria, you see the trace of this, they actually climb along the agar plate. This is an agar plate. On the agar plate, they are motile. They can move from here to here, but it's very slow. You can see the trace of the movement. So they are sort of like the slime layer, the trace of the slime layer can displace on their agar plate. All right. The last one, the extracellular layer, which there are three mentioned here. First one is capsule, second one is slime layer, the third one is so-called S layer. S stands for surface, right? There's no other meaning but simply just surface layer. And S layer present in archaea and also bacteria. But they are different. For the archaebacterial, you found this data is this S layer is indicated by this yellow color. So what are they? They are also called a protein jacket because they are coating on the surface of the bacterial cells. This look like a jacket. They're coating that on that a lot. And they can be either protein or glycoprotein. So in here, you see the tail here. This is the glycan, which is the carbohydrate. And the one at here, and the one that extending into the membrane is the protein. So this is the glycoprotein. And also this is another glycoprotein. And for example, here, this one, I think this one is the lipid. They can be in the form of part of it as a lipid, part of it is a protein, and part of it is a carbohydrate. All right? As you can see in the bacterials, gram-negative and gram-positive. So, you know that gram-negative and positive, they are very different in their surface. For gram-positive, here is the peptidoglycan. But for gram-negative, this is the outer membrane with the LPS. So, this S layer, for in the gram-positive, they actually adhere to the peptidoglycan. They have this secondary the peptidoglycan develop, and then they attach to this S layer. As for the gram-negative, it is not anchored on the outer membrane, but they are directly attached to the LPS. All right, this is the LPS. All right, so there is a presence of the S layer. I think I have described that, and it's also crucial for the pathogenesis. One example, always they use a mutant strain that do not cause disease, are sensitive to the phagocytosis and killing in serum, meaning that when they are tested, a mutant or the wild type, they see that the mutant always being killed by the macrophages. You can easily kill by our immune system. However, if they have the S layer similar to capsule and the slime layer, they are not easily being killed in the whole cells. All right. Another one is just a texture showing here. Not very clear, but you can actually Google and find a clearer picture. When this is the electron microgram showing how does this S layer, they arrange in a very pack. You see the very pack on the surface. So it's like this kind of structure. Now, what is the time now? It's already 1 hour, not yet, right? Shall I continue? Yes. Continue, we finish this part. Are you still okay? All right. Very descriptive. Every single structure is very descriptive. Now, this one, yeah, I think you're more familiar, is the external structure. There are 3 external structures we're going to talk about here. Pili or fimbriae or flagella. So pili and fimbriae slightly look similar, and flagella is very different from these two. Let's look at them. Fimbriae is something, the hair-like structure. They are very short, and they are made up with the protein. So the advantages, one of the advantages is that this fimbriae, they are very short, shorter than, if you look, you're comparing this, the pillars that connecting 2 bacterial cells. This pillars is very long, can extend to very long. But fimbriae is a hair-like structure, short normally, extended a lot out of the cells. If you look at that, they're about 1 to 5 micrometer in length and 3 to 10 nanometer in diameter. What is important is this one showing here. This fimbriae allowed the bacteria to attach to any surface, to the whole cell surface, or to the the metal surface, any surface. And because they have this kind of adhesin, which is also part of this protein subunit that make up of part of the fimbriae. All right. So first thing that you know that when the bacterial cells, they need to, they are normally free swimming bacterial, right? Individual free swimming bacterial, they can be easily be engulfed. Let's say they are in our bloodstream, easily be engulfed by macrophages. However, if they aggregate together, then they can easily survive. The thing is that before they can aggregate, they cannot aggregate while they are flowing in the water. They cannot aggregate when they are actually flowing in the bloodstream. They need to attach on the surface, any surface, tissue surface or any kind of surface. So attaching is basically the fimbriae can do that. This is the first step when they attach on the surface. And the second step, there are more. They actually recruit more of the bacterial cells on the surface, and then they aggregate on the surface. When that, they start to accumulate in a huge population. That's what the bacteria cause the troubles. Now, a little bit about the structure of the fimbriae. They have found, let's say, in the Salmonella type 1 fimbrium. This is one type of the fimbrium in singular, like this, one of this. In the Salmonella, what does it actually, they actually already identify each of the protein subunit of the, it's called fim subunit for the making of the fimbriae. And this fim actually have A, I, C, D, H, F, Z, Y, and number 4. Oh no, it's W, sorry. So this is actually in the operon, present in the genome. So they are actually formed as a cluster. So it's very convenient they form a cluster because they only require only one promoter present on the upstream. When they activate this promoter, all this gene will be expressed and transcribed. So it's very convenient, rather than individual actually controlled by different promoter. That's why it's called an operon, right? This is a gene cluster, they present, and therefore all of these actually showing here. Some of it is showing here, and these are those. So these are the fim protein, and each of the fim protein has makeup of its respective component. And they have this called sort of like making the oligomeric, sort of like chain together in a helical turn. And then they have this at the top, which is a hyaline, and it can actually attach to the surface, any kind of surface. Yes? The transcription process.
This direction or this direction?
All right, good questions. And is normally the transcription arrowhead is indicate these directions, but you do have some coming from that directions, right?
Yeah.
So that direction can be coming from another promoter, coming here. Yeah. All right. Normally how it's indicated, and it can be actually because it is double-stranded. Double-stranded, it could be one strand, they have a promoter here, this side. So from the opposite strand, they could have another promoter here, so they are transcribed in the directions. So, pili or pillars, pillars singular. So it has 1 to 10 per cells, not many. One thing you just need to know what is pillars do for the bacteria. It's also made up with the protein subunit called pilin. Right? What they're doing is very important because they do the mating of two cells, or we call it conjugation, bacterial cell conjugations. And I think you will be very familiar how conjugations are, what is the cells conjugations can do. Cannot see. This is one bacteria who has the plasmid, right? And this plasmid, very unique to the bacteria, but not all the bacteria have the plasmid, but this plasmid actually code for, cannot see, likely is there, it code for the pillars. So with this has the pillars gene on the plasmid, they can actually express and produce the pillars. So when it's produced the pillars, it starts to actually, when there is some other bacterial cells that around here, they actually can sense and produce pillars and make contact. When they make contact, right, what is happening is that this plasmid start to replicate. So when they replicate and then also transferring at the same times to the cells. So at the end, once this replication done and that conjugation is done, and the pillars will degrade it, and therefore it easily transfer a plasmid to another plasmid. And this only happen in bacterials and archaebacterials, yes, not a lot, and yeast, I don't think yeast has it. Eukaryote cells may not have it. So it's very unique because the plasmid, as we know, plasmid now we're using as a tool that we can insert any gene and to do any DNA recombination technique. So plasmid is one of the very useful, but plasmid is very, very important for the evolutions of the bacteria. Let's say if the plasmid contain the antibiotic, so the antibiotic can be transferred through DNA conjugation. So antibiotic resistant gene can be transferred from one to another, and this will become also antibiotic resistant. So it is very, very, it's kind of the way that the bacteria use to protect themselves. And what happened is that Neisseria gonorrhea using the pili to attach to the host cells, and it has been found. And attachment is very simple. Let's look at that. How do they produce the pillars and attach? So normally if they automatically attach, but then how do they produce? Now you have this pilin, one of these subunit of pilin, right? So this is the outer membrane, this is the inner membrane, this is the peptidoglycan, gram-negative bacterials, and this one here is the machine that produced the pillars. So this is another one, but this one doesn't have the filament, but this one has already the pillars already come out. So each of these pilin that make up of the pillars actually coming here. So they will express and produce and be transported to this machine, and they will be assembled through the machine, and they assemble and then into like a polymer, a very long polymer. This is how they keep coming and keep extending the pillars. So the way they extend and attach to the surface. This is how the bacteria can attach to the surface. And then how do they move? So how do they move is that they elongate the pillars, attach, and then they shorten the pillars. How do they shorten the pillars? Chop. They don't do that. They retract. So here is coming, they elongate, but they can also disassemble the pilin. When they disassemble, they actually start to retract and removing all the pilin subunit. So it's getting shorter and shorter when they retract. So therefore, when they retract, the whole cell's pulling. And then again, they come to a new positions and they extend the pillars again. You see how they move? They move in this way. All right? So they are not only used for conjugations, for mating, they use for motility as well as the attachment. Now, flagella. Flagella is totally different structure. They are they have the bacterials has the flagella, and they are very long. First of all, very obvious because they are long, and they allow the bacteria to swim because when they're beating and actually make the bacterial cells to move forward. And they are of course very large, 10 to 20 nanometer in diameter, and then 2 to 20 micrometer, I think it's about the cell size, in length. And first of all, we recognize that different cells, specifically, they have a different kind of flagellum. Now, therefore, they have a different name. If the cells has only a single flagellum, then they call monotrichous. If they have the flagella, many of them, but they are actually in one directions, from one positions, then they call rhophotrichous. If they have two flagella, one from the opposite end, they call ephitrichous. And they have the one that everywhere from the cell surface, they come out with the flagella, and they call peritrichous. So those are the table that describe each of the type of the flagella and the bacteria that having this kind of flagella. All right. And have you imagined how do they swim? If I have everywhere, this one is easily. If I actually rotate, right, this one single flagellum, I can actually beating and this moving forward, probably. But if you have both, how do they move? What do you think the bacterial cell will do? Of course, they are not moving left, right, like this. They don't do that. They have their way, all right? And I think I show you some of that in the next slide. So in here, very clever, if you only have one, right? If you have one, if they can actually rotate clockwise or anti-clockwise. This is anti-clockwise, counterclockwise. So this is anti-clockwise. When they rotate, they move forward. So when they do the opposite rotation, clockwise rotation, then they move backward. This is one type of the way they use. This is unidirections, so they can only rotate from the clockwise. So let the bacterial cells moving forward. Now, they only have one type of rotation, so that they need to stop, orient themselves into another directions, and then continue to move forward. So this must be slower. This is definitely faster. And about this one, the peritrichous, how do they move? Of course, when you have more than one, they're moving it very fast. You have the joy force of every single flagellum. So they're moving very fast. Now, when they want to change the directions, how do they do? So they try to separate all of this, right? Try to separate all of this, and then they're moving this and this, only of this, and then allowed to change the directions to here. So they are stopping moving some of the flagella, and then they beating some of the flagella, allowed them to move in the different directions. So when they actually already get the direction being changed, then they actually make it actually group them in a bunch, and then they move it at the same rotations, allow them to move in one directions. So they are not even they have a lot of flagella. They can control which flagella to rotate and which are not to be rotated. Looking at the structure, very simple. I do not need you to memorize this diagram. It's not necessary, but just want you to know there are three components that are for the flagellum. Now, the three components to making it is, one is the basal, so-called what? Basal structure. The first basal structure is totally embedded in the membrane. So this basal structure has 4 rings. So this is gram-negative. You see this gram outer membrane, peptidoglycan inner membrane has 4 rings. And these 4 rings, basically, they can rotate. They can rotate. When these 4 rings rotate together, actually is because they are all connected, right? They are also make this rotate same times. And of course, what is connecting the second part is the hook. The hook slightly curved. It's not straight. So when it is straight, I don't think you can rotate in the wider dimensions. So it is slightly curved, so it can rotate in the wider dimensions, so it actually can make it move faster. So it's slightly curved, and the hook are connected to the rod. The rod are actually inside the ring. Now, the last part is so-called filament. The filament basically is the flagellum that is the one to used for feeding. Okay, very simple three. So no need to memorize the structure, just let you know how they look like. This is gram-negative. For gram-positive, because it does not have the outer membrane, you remember for gram-positive, it only have the peptidoglycan and also the inner membrane, only have two. So for gram-positive, most of them likely only have 2 rings instead of 4 or 5 rings like the gram-negative bacteria. Right? The question is that all the time we're talking about the bacillus. What about the coccus? Do they have the flagella? Not many. Coccus, normally you don't find them have flagella, but they do found some of it have it. Enterococci, they have captured the picture. I think you see that. This is the coccus 1 and 2. This is 1, and they have the flagella. Very clear. They do have the flagella, but not many of them. All right. I still do not have my watch. Where is it? You want to have a break now? Okay. We have, see my time. 5-minute break. Oh, it's
ice cream. Oh, you use this one for? Can make it bigger. Yeah, it's today. Starting from next week. Yeah. But then you need to go up. Yeah. Today is 2 o'clock. Lazy mission lunch.
month you want to change 8%. But we have online, but we have 10 to 12 lecture physical, and if you want to do online, so I go to school, I have to. Oh. Thank you, George. Our Friday. That's Monday 24th. So at least Friday we're all open. Friday. Huh? Really? No, don't have lah. Just check.
What, anatomy? Lab? Sorry, I don't know.
On Monday, no? Lab ah? Really?
Friday night.
Oh, yeah, no. You guys got custody of the children. All three, all four. Right. You get cut. From room.
Really? You asked me, I asked
Oh, you know why? Because they take my chemistry to 9-12. You also chemistry? Chemistry 9-12 Friday night. Yeah, 他们去到9-12. Yeah, between Friday to Tuesday.
Friday 啊？
可是他讲你是为了专业，我看
他是我。Oh, very bad. Did you what?
But it's nice, Sam.
You don't have to do it. Yeah. Yeah. No. Take already. Yeah.
purposely put one question, but each one time I support everybody. So it's like you send to school, what?
Yeah.
Like,
But our deadline also this week. The end of the month.
Thursday.
Yeah, that's what Dylan, he said no, if no use, no can. Because he said what he said. You don't have to find
Alright, just make some announcement. Tomorrow we have, tomorrow and day after tomorrow, Wednesday, Thursday, we have the practical class. Okay, you know the rule when you enter the laboratory, so you must have a lab coat with you. I think some of you may not, new student may not have the lab coat. You can borrow from the lab technicians, but make sure that you should buy your own lab coat in future as well. And second, you should have the cover shoes with you. And is lady, you may have to tie your hair because we're going to turn on the Bunsen burner. I don't want you burn your hair, right? And what else? It can be very long, some of the practical class. But the first one should be okay. But the second or the third, I don't remember, second or the third, it could be very long. So try not to be freely go to any practical class as you like, especially though the one that on Wednesday afternoon is already in a full capacity. Not allow anyone to join the Wednesday 2 to 5 if you are not in this group. All right? But let's say that if you want to change for any reason, very valid reason, you want to sort of like attend the other group, you are allowed. Better than you are not attending any lab practical class, because each practical, the lab report, I think will be counted in your final mark. All right. Also, you are allowed, if you are hungry, you are allowed to eat in the class, in this lecture hall. Unless the lecture hall, the person who hold the lecture hall say that you cannot, but otherwise I'm okay. Just not to make noise, that's the only thing. All right? You also can go in and out freely. Either, I do not know whether it's back door or front door is also, you can go in and out to washroom. It's okay, no need to ask me. Just quietly walk out, yeah, and come back. That I feel comfortable if I'm sitting here. All right, we continue. Not very, what kind of speed? 2 times speed to go through all this? So this is the intracellular organelle from the bacterials, which we're going to go through this. Cytoskeleton, intracytoplasmic membrane, inclusion body, ribosome, nuclear, and plasmid. Very quickly, because some of those I think you're quite familiar. Anything happen there? All right. Now, first thing, that's cytoskeleton. All cells must have cytoskeleton. Without cytoskeleton, you cannot hold the cytoplasm. Any cells, eukaryote, prokaryote. But eukaryote and prokaryote cytoskeleton actually making the intrastructure, the infracellular structure of cells, they are different between eukaryote and prokaryote. Here, they have already identified a few which is quite common and also homologous to the eukaryote cytoskeleton. Now, the first one is the called physic. Physic, they found the physic is homologous to tubulin. We know that in eukaryote, there are 3 very basic cytoskeleton that present in the cytoplasm. They are tubulin, microtubule, or the second one is the actin, or they call microfilament or actin filament, right, in eukaryote. The third one in eukaryote is called intermediate filament. So they all find these three basic cytoskeleton also present in the bacterial. And the first one, which is homologous to tubulin, and they form the zig ring. The fifth zig, actually, during the cell division, they are present as a ring. That's why they call zig ring, and the center of the cells. This fifth zig would develop into the septum. The septum is the plate that dividing the cells into two dorsal cells eventually. So they are actually indicating a cell division happening on the zig ring. The second one is the homologous to the eukaryote microfilament, or called actin filament, and they call MREB. So this is also a protein, MREB. And MREB, the zig ring is at the center, only a single ring. But MREB, you found that is just at the peripheral regions, underneath the membrane. They have this growing of the MREB. Right? They actually holding the structure of the cells. And important, first of all, holding the structure of the cells. Secondly, they are moving the chromosome to the opposite pole. So important in cell division when they're actually transporting, okay, the chromosome into the two ends, separate the chromosome into two. And what do I mean by holding the cell shape? This MREB, when you look at it, they only present in the bacillus, in the rock-shaped bacterial. In the coccus, they do not have MREB. So this MREB, they may have something similar, but they do not have exactly MREB. This MREB is holding the bacterial cells as a rock shape. If you without this MREB, they actually can be in a spherical shape. Right? This is important for holding as a rock shape. And finally, the last one is the so-called chrysanthemum. I think I mentioned that in do not know which chapter, last chapter or the one before. Chrysanthemum has a homologous protein in eukaryote called intermediate filament. As we know that intermediate filament is also made up with protein, but are very rigid. When the intermediate filament form in the hair cells, in our hair cells, we also have this kind of intermediate filament. They make the hair become rigid, not easy to bend. So therefore, we know that intermediate filament is for the rigidity. So same thing that present in the bacterial cells, during the cell divisions, and you can see that this chrysanthemum actually expressed at the one side, and you're actually present at one side and detected by this fluorescence microscopic picture. So they are actually present at one side, and because of that, if you remember what I mentioned earlier, because of that, they actually holding this side and not allow the peptidoglycan to grow at this side. So this side is sort of growing very slow, the cell wall. However, for this side, without the chrysanthemum, they grow much faster. So because this side grow faster and this one grow very slow, eventually you have a band on the shape of the cells. So you have a chrysan-shaped bacterials because of this, the presence of the chrysanthemum protein. All right. So these are the basics cytoskeleton for which involve greatly in the cell divisions. And the second is the intracytoplasmic membrane, ICM. This is not commonly seen in all bacteria, but you will find them in the proteobacterial, or called the purple non-sulfur bacterial. So the example is those the Rotobacter, Rotospirums, and Rotopseudomonas. And what is ICM, this intracytoplasmic membrane? They actually, in a way, because they are doing the photosynthesis, all right? They invigilate, meaning that the membrane, the cell membrane, inside, they actually invigilate, growing into the cytoplasm, and then they've been folded into many layers. So these ICM are actually originated from the cell membrane. and they just invigilate into the cytoplasm and growing layer and layer. Why do they need this layer and layer? And because of the photosynthesis, they will have a, because in this this ICM, they contain the chlorophyll. I think this is Wikipedia, and they have the chlorophyll. I think where do I see the chlorophyll? Which part show the chlorophyll A and B? Do they mention that? They contain chlorophyll that actually can do the photosynthesis. As you have a larger area, and you can absorb more light for the photosynthesis. That's why they have this kind of layer of membrane. So this two is only showing different type. This is actually in this way, right? And this is another way. So this is the EM picture, and this is another EM picture showing these are difference. What else? This is different from cyanobacteria because cyanobacteria has thyroid, which is quite distinguished from this type of ICM, right? But they can, remember, this proteobacterial, they can actually do photosynthesis. Inclusion body. Inclusion body, as you look at it, is like something like our fat cells. It's the granule that actually like this. For our fat cell, we have a lot of store a lot of like fatty acid, but in this granule, they are for the bacterials. They are store organic and inorganic materials. And why they need to store all the nutrient? Actually, to preserve when they come to a tough time. So, what are inside this organic and inorganic? They are polybeta hydroxybutyric acid (PHB), and glycogen, starch, lipid, hydrocarbon, and cyanophysin. So those are phenolcycin is a compound that this cyanophysin, especially present in the cyanobacteria, so that's why it's called cyanophysin. It has the structure contain a lot of nitrogen. So they think this particular organic is to the storage of the nitrogens. So, and what they have found is organic and inorganic materials, and especially when they start to produce a lot of this granule is during when the cells grow. You know the cells grow, right? When they grow to a certain stage, there is a depletion of the nutrient, let's say. Then they will go for the stationary phase. There's no more growing. So at this stage, if there are no more nutrient, they die. They will drop, right? So during the stationary phase, how do these can survive? One way is to produce a lot of these inclusion body and for the storage during the stationary phase, so that they can allow them to actually pass through. But of course, if you culture them in the lab, definitely they die because there's no more the supply of the nutrient in the medium. And this is how they actually store all they're actually in the form of polymer, right? And one more thing is that they are formed as a polymer is because if they are stored as a soluble material and not polymer, they actually create a lot of osmotic pressure inside the cytoplasm. That's why they actually form as a solid polymer and they reduce the pressure inside the cells. If you have storing a lot of water in this granule, right, eventually the cell may burst. Okay, the next one, other inclusion body. This is interesting because this I think is present in those bacteria, actually aquatic bacteria. I think cyanobacteria may have it. They actually look like this. You see this one? Yeah, they are calling, what is it calling? They are vacuole. So in this vacuole, there are small vacuole. They are in this cross section. When you chop it, the cross section look like that. If you do a longitudinal sections, you will be like this, right? A line like this. So this is longitudinal section. This is a cross sections. So in this very big, this is a gas vesicle. Gas vesicle, as is what it means, is storing of the gas. It's the storage of the gas. So they are, when you look at the microscope, they are a little bit shining. but not like this, and then easily be detected. And actually it occupies a lot and almost the entire cells. And they are important because in some of that, for example, cyanobacterium actually undergoing photosynthesis. So sometimes they need to flow at the surface of the, let's say, of the ocean. They cannot all the way sink to the sediment. So they need to flow. This gas vesicle allowed them to flow. Right? Because inside they contain the gas and then they allow them to flow. And more important, how the gas being stored in this kind of vacuole, and because the materials that making this vacuole is hydrophobic. So inside is really hydrophobic. If it is hydrophobic, it wouldn't allow the water to go in. So the hydrophobic structure is just prevent water to go in. If water go in, then it will sink. The bacterial cell will sink. So it's only allowed the gas to go in and allow it to flow on the surface. All right. Ribosome. Ribosome, everyone's familiar. Ribosome has a lot in, very easy to be detected. If you look at the bacteria under the EM, you will have the black color. The black dot here must be the ribosome. Did they point it? Yeah, they pointed ribosome. And of course here is the nucleoli, which is the white, and the dark normally is ribosome. Ribosome, 70S. In bacteria, ribosome is 70S. That is consists of two subunits, which is 50S and 30S subunit. And this, what is this ribosome? How to say? Ribosome is contain not only the ribosomal RNA, is the ribosome contain ribosomal RNA plus a lot of protein, complexes of protein. And that's why when you're actually thinking that, oh, this 70 and this 50 plus 30 should be 80, but it is not. Because this is also, what is the unit, the S stand here, the stand? Anyone? It stands for Sibber. Is actually is sedimentation coefficient. It's just a unit, right? About the density of the ribosome. So you have further, the large subunit can break down into 23S to 5S, and then further there is also a small one is 16S. What about, let me ask you a question. Antibiotic can target the bacterial ribosome, right? So we take the antibiotic, that biotic target the ribosome, destroy the ribosome, bacterial ribosome. And how come the antibiotic doesn't destroy the human ribosome? We also have ribosome, right? In our cells. Why it doesn't destroy the human ribosome? Anyone? And the reason is that, anyone? We have a different ribosome from a bacteria. Our ribosome is not 70S. Our ribosome 80S, if I'm correct. 80S. Our 80S ribosome can break down into 60S and 40S, big or small. You see the ribosome structure, right? Big and small. So we have a different kind of ribosome. It's not the same as the bacteria. That's why the antibiotic can target of the bacterial ribosome and destroy it and stop the translation, but not for human cells. A lot of ribosome, 20,000 ribosome per cells, and then use for protein synthesis and for translation. And don't think that I need to explain this, what is translation. I think you should know. Right, the last one is the nucleoli and plasmid. Always remember, nucleoli is actually the chromosomal DNA of the bacteria, and it's called nucleoli. It doesn't have a nuclear membrane. It's not like eukaryote. Eukaryote have a nuclear membrane that cover the DNA, the chromosome, but it doesn't actually is sitting at a particular regions And this region we call the nuclei, including the chromosomal DNA. And what else? Just additional. In addition to that, we also have the plasmid. Here say, I don't know whether it's 2014, it's not very updated. Here also, I don't think it's very updated. So it's already complete sequence of plasmid. Plasmid is something that additions for the bacterial, but it doesn't require for bacterial. Plasmid present in bacterial is maybe serve as a purpose to produce the pillars, right? Or maybe the antibiotic resistant gene, but it doesn't require for bacterial. Fill in the blank. Plasmid does not require for bacterial. Yeah? It doesn't require for bacterial growth. Without the plasmid, the bacteria survive happily. So it's something that, therefore, it can be transferred to others. It's something that additional. So the plasmid can be 1 kb to several kb. And as you know that the plasmid is also can be used as one of the, as I mentioned earlier, can use one of the two for the recombinant DNA technique. This is just a diagram. This is just a diagram of the plasmid. They are actually turned into a supercoil. They can actually turn in a supercoil form. Have you run a plasmid on a gel, agarose gel? No. It's fine. Because when you run in a gel, you can actually see them, they are in a supercoil. That is an indication they are in a supercoil. Alright, plasmid, as I mentioned, can be passed from one bacteria to another bacteria. If something have an advantage for this bacteria to survive, if they have it depends on what is being coded, what is present on the plasmid. So there are 5 types of plasmid here. The cell replication, I will talk about that later. So there are 5 types. Fertility F plasmid. So they produce the pillars as the one that having here, they can produce pillars and mate with another cells. And resistant plasmid, which is the antibiotic, meaning that the plasmid, they have the coding gene for antibiotic resistant. Virulent plasmid, this is actually present in the bacteria that affect, the agrobacteria that affect the plant. And you have coplasmid, which is also produced bacteriocin or colicin. Bacteriocin can be produced by bacteria, but this bacteriocin is basically also killing other bacteria. So between the bacteria, different type of bacteria, different genus, they can competitive in terms of surviving in the environment, in terms of like securing the nutrient. That's why they themselves can produce bacteriocin and kill other bacteria. So you have the plasmid can get enzyme that also can produce to digest some like xylene, very difficult to be digested by normal cells. So they have specifically produced something can survive in the very certain tough environments. So all this is the 5 type of the plasmid. And what I mean by cell replication during cell divisions. Plasmid, as I mentioned, it doesn't require for the growth, right? So during the cell division, either it will replicate, but it can be lost as well. So when the cells grow, they can simply losing their plasmid when they grow. So ending up, they can gain the plasmid by doing the conjugations, but as they grow from 1 to 2, 2 to 4, those plasmid can easily lost. Just maybe one daughter cells will have 2 plasmid, the other daughter cell have none. Although the plasmid can replicate. So remember that they can be lost during the growth, and they can gain the plasmid during the the cell mating. All right, this is the last, just to show you the diagram. Where is the bacteria? This is the nuclei, the bacteria nuclei. Yeah, during the cell division, so they are separated into two, and with equal amount the separated these two chromosomal DNA. Now we come to the very last part, which is the endospore, the development of endospore. One thing you want to remember, which bacterial, common bacterial, they have the endospore. Not all the bacteria have endospore. E. coli has no endospore, but the bacillus, the very common bacillus and Clostridium, they both have endospore. You may have just remembered these two. For the other one, Sporosuccina, not very common that we normally mention, but for Bacillus and Clostridium, always remember they are gram-positive. They making the endospore. Why you need the endospore? Because the endospore is very tough. Is considered a sexual reproduction, sort of a sexual reproduction for the bacteria. We know that bacteria is undergo a sexual reproductions, like by cell divisions. But sometimes we consider endospore for sexual reproduction. All the bacteria up to the very simple eukaryote, yeast, or even animal, when they do the sexual reproductions, is actually allowed them to survive under a very tough environment. This is one thing. So producing the endospore is to allow the endospore to pass. For example, in winter come, the temperature can go under like minus 40 degrees Celsius. How the bacterial cells survive? They may be frozen and right, be broken, right? So endospore allowed them very tough to actually allow to survive under minus 40 degrees Celsius. So therefore, they are somehow evolution is amazing. They are making this to keep the cell alive. And when the times come, when you have water, nutrient, and the temperature increase, and then they germinate and become the cells. So that's why they actually survive in this kind of condition: desiccation without water, high temperature, and also UV radiation or chemical. Chemical, we can consider antibiotic as a chemical. Now, talk about how they actually make the endospore. Step by step. This is called vegetative cells, just a normal bacterial cells. Right? We only call it vegetative cells when the cells, they are producing the endospore. So we call it vegetative cells, just a normal cells. So when the first step they start to make endospore, they actually start to replicate the DNA, chromosomal DNA, into two. So it's just like very normal cell divisions. And then they actually separate into the two end, and then they will have a divisions on like into two daughter cells. However, there is only one daughter cells will form the so-called four spore. So how do they do that? So the other one, this chromosomal DNA from the other one will be degraded. Only the four spore, the chromosome will be preserved. And what happens is that there is an invigilations, meaning that engulfment, so from the membrane. So each cell has already, as one daughter cell, they has one membrane. So when there is a coming here, this is extending here, so they wrap around this four spore. When they wrap around, then they produce a second membrane. So it's actually double membrane, right, under this four spore. What happened next is then they're forming a so-called cortex. Cortex is containing peptidoglycan. It's containing peptidoglycan. So they deposit the peptidoglycan and form the cortex. At the same times, there is also the picolinate acid and also calciums also deposit into here, into the false spore. So these two actually form something that protecting the DNA, which I think I can mention that later on. So when they're actually forming the cortex, the next one is forming even the outer layer, and we call it spore coat. Right, the spore coat they have, and then from a spore coat they have inner, and then even there is another one, outer spore coat. So all of these has form a layer. One, they actually mature, and these we call parental cells, or we call mother cells, will be damaged, or it actually breaking it out from the parental cells, from the mother cells, and then it becomes an endospore. So this is how the process, all right? And if you're looking at the people actually analyzing this particular structure, the endospore, they have many layers. And each layer, they serve a purpose just to protect the DNA, the chromosomal DNA inside the spore. So if you look at that, they have, starting from outside, I don't even remember what are they. Sorry. They have the very external, they call exosporium. They are formed by protein or glycoprotein. They are protecting the spore from any chemicals. All right. They also can adherence because they are the outer layer, and they are the protein. Protein normally can stick to surface. Now, they have the outer coat and inner coat, the spore coat. This is keratin. They make up keratin, which is, I think, is quite tough structure. And they have a lot of disulfide bond, meaning that the structure is very tough because the disulfide bond forming in protein forming the double bond. So they are actually very solid, very firm and packed amino acid in the keratin. So they're resistant to what? To UV radiations and chemicals, right? This outer phosphor membrane is said to have the lipid and protein. So it's become a permeable barrier. They not allow any chemical to go in. So for this one. Now, furthermore, this cortex, so it's a very thick layer because very obvious under the microscope. It contains peptidoglycan mainly. So it protects the inside here. Everything is trying to protect the chromosomal DNA. Inside here from the chemicals and from any dehydration and also the heat resistant. Germ cell wall, they found another layer, but they are not too sure whether this is a peptidoglycan. And eventually when they become individual cells, the vegetative cells, and they become the cell wall of the vegetative cells. And inner phosphor membrane, here, right, the inner layer here. This is also lipid protein similar to this. This is the outer phosphor membrane, outer and inner. So they are also impermeable for the chemicals. The core itself, it contains a very large amount of calcium with the dipicolinic acid, and it protecting the chromosomal DNA from resistant to the heat and UV light, especially the gamma radiations. In addition of that, these two calcium and dipicolinic acid can protect the DNA. This also additionally, they have this so-called acid-soluble DNA binding protein, when they also binding on the protein to further protect the protein from any damages. For example, from the gamma radiation, because the gamma radiation can break the DNA backbone. UV light can change a single base of the DNA. It can actually reverse back, let's say. But breaking gamma radiation is further causing more damage by breaking the backbone. So binding of this DNA binding protein on the DNA is further protect the DNA from breaking up into two pieces. So all of this, too many layers, but what you need to keep in mind is this, the spore coat, And forget about this. This don't even know what is this. And then the cortex is important. Of course, the core, what is inside here, and also these two. So these two, cortex and also the spore core, and the one that you might have to keep in mind because they are being very well studied. All right. So this is the very last slide. Excellent. Okay, that's all for today. Any questions that you can raise now if you have any questions?